Louis, I'm the CEO of Treefrog Therapeutics. As you can hear, a French-based company. We are based out of Bordeaux, which is more known for grape fermentation than cell therapy, but hopefully that will change. We are focusing on regenerative medicine, and the backbone of the company, as most of the company we've seen today, is a technology. So I will spend some time on the technology just to give you a highlight on why we think we're special, and then I will dig into the different programs we have. You'll see we are a very focused company, so we have only 2 programs, so it won't be too long. Okay, so we have a unique technology. Clearly, we are disrupting the way people see the manufacturing of human cells. And we believe, very bold, that this might really break the glass ceiling of cell therapy and will allow for the mass production of human cells. The key of what we're doing is 3D biology. You will see that everything we do is around microtissues, 3D biology, and trying to understand how we can scale this 3D format. And we have some evidences, but I won't go into too many details today on what it brings to the patients in terms of safety and efficacy. Then I think we all have to remain very, very humble in terms of what's happening today in this marketplace. I think we are learning a lot. We've been learning a lot over the last years. The company is still young. We've been learning a lot, and we are still investing a lot in our technology and the understanding of 3D biology. I think we also need to learn more about how to process cells to get better consistency in what we do. And I will show you what we obtain in our lead program in Parkinson's disease, which is, I think, really something really new to the field. Okay. Okay, so what is system? So here on top right, you see iPSC cells growing in a capsule. In fact, our technology is pretty simple. We protect the cells from the manufacturing process by putting them in a capsule of alginate, so very simple technology, very thin capsule of alginate, so it's an empty capsule. And so you saw the movie once. And the particularity of this technology is that when we mimic the biomimetic environment in the capsule, the cells are growing in a sphere format, so in a monolayer of cells. And we think that brings a lot of the benefits we see with the technology. And you see on the second one, so the running movie, that we can now encapsulate cells at a very, very high speed. We today are encapsulating cells of something like thousands of capsules, a bit more thousands of capsules per second. So it's a high throughput technology. And it's fully scalable because when cells have checked into the capsule, they don't care if they are in 5 ml or 5 liters or 5,000 liters. So the question is how you encapsulate them fast enough, and then how you manipulate them or how you process them when they're in a capsule. And then I don't have a video today, but we can also remove the capsule very easily and get access to the cells directly. On the bottom right, what you see is the encapsulator. So that's our current machine that is designed to go to up to 10-liter bioreactors mainly. We are bioreactor agnostics. Our technology is really putting cells in capsules. The rest is the choice of, or the need of the project. And what you see then on the right is more like in numbers what it means. In fact, when cells are in this biomimetic environment, they don't die, or they die very, very little. And we have a very high survival rate, which explains why we have also a very high amplification rate of our cells in our technology. We know they don't die because they stay in the capsule, so if they die, we see them. What we have measured, so we all have numbers, but what we have measured is that when we grow cells in 2D, so in plastic dishes, we usually lose between 40 and 60% of the cells in a week. In our case, as you can see, it's less than 2%. And obviously, the quality of the cell is there too. So what do we do with that? We do special things. So I will show you the two main examples we're working on today, which is Parkinson's disease and acute liver disease. So Parkinson's disease, why that? First, because there is a very important unmet need. Patients are progressing. It's a very progressive disease, and you know this disease because you're in the field. There is no solution besides surgery at some point. And the new promises of cell therapy based on iPSC is to be able to relocate dopaminergic neurons in the striatum of patients. And the need is huge. When we look at the competition in the market, we don't even look at it as a competition, but as a cooperation, because there will be very little actor in the sector being able to serve this market because it's really big. What we bring here is a new paradigm. We bring a product that we think is a best-in-class profile with a very strong efficacy, with a lot of better safety, we think, than what is done in the competition, but also with consistency. We are running today in the lab batches of manufacturing every other week. We are already running the batch number 45, 46, and we have a very high consistency. When we looked at the key parameters of our product today, the error bar is below 10% in terms of critical parameters. So it's very, very consistent technology. We have done a lot of proof of concept studies with our product, 4 of which have been finished, and there is another one going on. And we are starting to interact with the FDA and EMA to evaluate the product, and we're on track to start animal studies and file for IND early 26. We are developing a product in preclinical with one aim, is to bring to the authorities the final product. So we're taking our time before we go to animal tox studies because we want to make sure that we won't change anything. So the product we are today presenting to the authorities is the final product, is the commercial product. And that's the first time, I think, in cell therapy that we are, in allogenic cell therapy, that we are in such a position to go to the authorities saying, this is the product. There will be no scale up. There will be no change in the process. This is the final product. So what we've shown with this product is that, first of all, we have a long-standing efficacy. Today we have looked at 8 months. Hopefully, we will have longer data soon. We can cryopreserve our product, so it will be an off-the-shelf product, obviously. And as you see in the middle, in fact, our product is a macro tissue of brain cells containing dopaminergic neurons. So when we graft them, they already generate dopamine, and we bring efficacy, and we bring short-term efficacy, and we see that very short-term, we see an impact of this dopamine released in the brain of rats today, obviously, hopefully humans in the future. And we also see, and it's difficult to assess with like cutting data, but that we see a much better survival of the graft, of this 3D object, than when we inject single cells. So it's difficult to assess because it's a very difficult parameter to measure in terms of when you are in the brain of an animal. But anyway, we have a very strong feeling that the dose we inject is the dose we see in the animal, and there is a good linearity in terms of dose effect. We have a specific product, very differentiated from competition, and we can release our batches by measuring dopamine, for instance. So we have a real, let's say, COA for many parameters. We have shown that the graft integrates very well, and as I said, there is a very high consistency, not only in the manufacturing, but also in the way we deliver the dose. So we think there we will bring certainly some elements of safety for the patient also by being able to really measure the dose we deliver. In terms of production, as I said, we already are at commercial scale, and we have a very high consistency in the production. Okay, the second program we started about a year ago, so it's still early. It's in liver disease. Why we did that? Okay, if you've seen the other presentation today, there are other players in the field, so I will not go into too many details on why it's important to bring an off-the-shelf product to patients. But off-the-shelf mean frozen, mean accessible, and mean also that you have the product. Today, it is accepted in the field that you will need something like 5 billion of hepatocytes per patient if you want to have an effect. So that means that if you want to make a batch for 100 patients, you need to be able to produce in one batch 500 billions of cells, of human cells. Nobody can do that today. We cannot do it today. Today we can do maybe 50 billion, but we're not there yet for the 500 billion. We're working on it, and we hope we will be there in the coming years. But it's a big challenge for the industry, and if you're not able to produce those cells, you will not have a product. So it's a big challenge. We are there. We have a product, well, we have a candidate that has the features we want, the efficacy we want, and that we think we can scale at the scale we need. Now we need to show it. So we'll show that hopefully in the coming years. So our model is very simple. I didn't insist too much on the technology itself, because technology is a mean to do something special. And I think we are doing special products, and we'll keep on doing that. But we need to remain very focused on where we invest. So you saw Parkinson, you saw liver. Those are the two areas where we want to work. Those are proprietary programs. But at the same time, we are in discussion to out-license our technology to partners that will need this technology to scale their manufacturing. And we are happy to, obviously, bring our technology to them for patients, obviously, and also for our shareholders to create a bit of value, as you know. But clearly, this dual tech strategy is working very well, because it gives us the means to really push our internal programs. The third leg of the company is technology. We are a very technology-driven company, and we will keep on investing in technology and technologies, actually. We have developed over the last 18 months teams in analytics, of course, but also in imaging. We have a very strong team in imaging now, because we believe that's one of the key elements to understand what you're doing when you grow cells, when you make cells, to look at them and to look at what's happening. But also in gene editing, and we are piloting a series of technologies to serve our markets. So we're developing very fast. We are now 125 people, 10 people in Boston, the rest in Pessac, in Bordeaux. I am a very lucky CEO. I have a very strong team. Most of the people on this slide, besides the two founders, Kevin and Max, joined the company in the last 2 years. So we are really on the move, moving very fast. And if you read very well the Lavrock slide just before, we have something in common with Lavrock. I see you smiling. Yes, Elsie Boglioli, my chairperson, my chairwoman. She's also working for Lavrock. She's great. We have a great team. We have a great board, very engaged. We have a very dynamic company, and we are looking forward to bring new things to the field and to help some of the other players to really be successful, because again, with just the beginning of cell therapy, we have so much to do all together that we really think that we should really work more in teams. With that, I'm finished, and thank you for your attention. And if you have any question, otherwise, during the break or tonight with a glass of wine, it'll be a pleasure to answer your questions. Thank you.